Hello everyone, Biochemistry channel is here with the topic titled Amino Acids, Peptides and Proteins-I. Please visit biochemia.web website for a more comprehensive document of the topic in PDF format. You can find the link in the description. I wish it will be useful. Let's get started. Here is the topic content, the overview. We will start with the overview of the topic, and then the major title is Amino Acids for today. We will talk about the classification and continue to the end chemical properties. Proteins mediate almost every process that takes place in a cell and exhibit an almost infinite variety of functions. Proteins constitute a very important part of the living organism. And the word protein means leading in Greek. They carry carbon, hydrogen, oxygen, and nitrogen in their structure. Growth, reproduction, and self-repair, which are the main features of life, are closely related to proteins and nucleic acids that form very important compounds of proteins. We biochemists almost inevitably study one or more proteins to discover or evaluate the molecular mechanism of a biological process. Proteins are the most abundant biological macromolecules found in all cells and in all parts of cells. Proteins are also very diverse. Thousands of different species can be found in a single cell. Plants synthesize proteins from compounds such as carbon dioxide, water, and inorganic nitrogen. Animals cannot do this. Animals depend on plants, or animals depend on other animals for their protein requirements. Although there are amino acids, the amino acids, which are the building blocks of proteins. So some amino acids are essential, but some amino acids can be derived from other class of compounds in the metabolism. But in general, almost half of the amino acids, half of the proteinogenic amino acids are essential for animals. But plants can synthesize the amino acids from carbon dioxide, water, and inorganic nitrogen. Proteins are large molecule substances. Their shape can be fibrous, oval, or globular, or spherical. Their building blocks are amino acids, and the proteins are amino acid polymers. The genetic code determines their synthesis. Relatively simple monomeric subunits, which are the amino acids, provide the key to the structure of thousands of different proteins. In the synthesis of proteins, 20 amino acids are used. The proteins of every organism, from the simplest bacterium to man, are built from the same 20 ubiquitous groups of amino acids. Since each of these amino acids has a side chain with its own unique chemical properties, these 20 groups of precursor molecules can be regarded as the alphabet in which the language of protein structure is written. While half of the amino acids used in protein synthesis are synthesized as intermediate metabolic products, and some are required to be taken with nutrients. This means they are essential. But for the plants, they can synthesize from the simplest building elements for their amino acids, but it's different in animals. Protein molecules contain about 45 to 55% carbon, about 6 to 8% hydrogen, 20 to 25% oxygen, and 15 to 17% of nitrogen. And the nitrogen, it's a special element for protein, or it looks like a special element for protein. It's found in about 60 as a mean value, 60% of average molecules. And based on this determination of the amount of nitrogen, so we can calculate the amount of crude total protein. And the method we can use to measure the nitrogen content of a given material, the biological material, it can be animal source, maybe blood serum or any tissue. it may be also a food substance. So the method is Cage-Dahl method. Let me write for you the Cage-Dahl method, so you can check via web to take more information about the Cage-Dahl method, that we can use it to measure the crude protein by measuring the total nitrogen content. Many different proteins are found in different tissues. Each protein synthesized in the body is structurally and functionally unique. They participate in processes that characterize the individuality of cells, tissues, organs, and organ systems. A cell contains thousands of proteins, each with a different function, such as enzymes, and every reaction in living cells requires enzymes. Transport proteins, such as lipoproteins, transfer in bilirubin-binding proteins, storage proteins, such as myoglobin, defense proteins, the proteins which have function in coagulation or immunity against pathogens. These are the immunoglobulins or antibodies. Contractile proteins, actin myosin or the muscular proteins, and structural proteins, the connective tissue proteins, collagen and elastin. And here's a table showing the protein ratios in some tissues and also foods. And also it's a kind of comparison with the foods originates from animal or the animal sources foods and the plant foods. And pay attention please about the percentage, protein percentage of meat and percentage of beans and soybean. And as you see, the soybean or the bean has more content of protein than the meat. However, the meat especially is important for the animals because meat contains more branching amino acids, which are the essential amino acids, than a plant protein source. Amino acids. We will look to the classification of amino acids. Then I will introduce you one by one, especially the proteinogenic ones, and then the modified non-protein amino acids and essential amino acids. And at the end, we will look to physical and chemical properties of the amino acid. Proteins are polymers of amino acids, with each amino acid residue linked to its neighbor by a specific type of covalent bond. Proteins can be broken down or hydrolyzed into the constituent amino acids by various methods, and early studies of proteins focus on the free amino acids that are naturally derived from them. Now we have information, we have knowledge that 20 different amino acids are commonly found in proteins, thus we call them proteinogenic amino acids. Compounds with amino and carboxyl group in the molecules are called amino acids. But the name is derived from the two functional groups they carry, the amino group and carboxyl group. In the structure of amino acids, 4 different groups are attached to the 4 valence of carbon. So these are the carboxyl group, amino group, a hydrogen, a single hydrogen, and an R group, a radical group or an unordered group, actually, which give the unique identity of each amino acid. So it depends on the R group, maybe an aliphatic chain, heterocyclic chain, or an aromatic chain that we will look. The different kind of amino acids are derived. Asparagine was first discovered in 1860. Threonine, the last of the 20 amino acids discovered, was not identified until 1938. All amino acids have trivial or common name, in some cases derived from the source from which they first isolated, such as asparagine was first found in asparagus, and glutamate in wheat gluten. Tyrosine was first isolated from cheese, and the name derives from the Greek word tyros, which means cheese. And the glycine derived from the Greek word glycose, which means sweet, because its taste is sweet. Amino acids are indicated by a single or three letters. For example, the glycine, the three letter of glycine is the GLY, or one letter is the G. The three-letter code is transparent. Abbreviations usually consist of the first three letters of the amino acid name. The single-letter code was developed by Margaret Oakley Dayhoff, who is considered by many to be the founder of the field of bioinformatics. And the single-letter code, as seen here, for the G for glycine, reflects an attempt to reduce the size of the data files used to describe amino acid sequences. The carbon atom closest to the carboxyl group in amino acid is called alpha. And let's look at the core structure of an amino acid, which all share a common feature, which they have a carboxyl group as seen here, and an amino group, and the hydrogen attached to the alpha carbon. So this is the alpha carbon, thus which makes the hydrogen alpha hydrogen, and the amino group alpha amino group. And the amino acids having this structure are called alpha amino acids. This is important because also there are beta amino acids. And the R represents the radical group, or the group that the amino acids differ. And if the amino group, assume there is another carbon here, and the amino group attached to the third carbon in the chain, this is the first carbon, carbon, second carbon is alpha carbon, and beta carbon is the beta carbon. If we have a molecular structure having amino group with the third carbon, thus we have the beta amino acids. And beta amino acids can also present in different species, or they can be biologically important molecules, such as in the field of drug design, biological research, or maybe for bacteria or antimicrobial peptides. But for the proteins of organisms, the proteins of mammalian or higher organisms, the alpha ones are the important. In here, there's a table showing 20 proteinogenic amino acids and their both three-letter symbols and single-letter symbols for you that you can check when you want. Amino acids are linked to each other by acid-amide bonds called peptide bonds to form protein. So the acid-amide bond, we learned that kind of bond also in the phospholipids, but here the acid-amide bond is named privately as a peptide. This means if we have an acid-amide bond formed with two amino acids, it is called peptide bond. And here is the peptide bond, which means when an amino acid join with its carboxyl group and another amino acid joins with the amino group, so this is a condensation. This means 1 mole of water is removed, and acid-amide bond is formed. Acid-amide bond is formed. Especially it is called peptide bond. Let's look at classification of amino acids. Amino acids are classified in various forms according to their properties. For example, they can be classified according to the number of amino and carboxyl groups in their molecule, such as monoamino monocarboxylic acids, monoamino dicarboxylic, diamino monocarboxylic, diamino dicarboxylic acid. Another classification is we have an aliphatic amino acids, which they carry aliphatic side chains. The side chain means the R groups, which they can be neutral, acidic, and the basic amino acids, and sulfur carrying amino acids, the cysteine and methionine. a good question, maybe a good question for you, such as which of the following is an amino acid carrying sulfur group or carrying phenol or phenol ether group? And aromatic amino acids, the amino acids carrying the benzene ring or the benzene derivative ring. These are the tyrosine, phenylalanine, and the tryptophan. And also there's a heterocyclic amino acids, which there is a cyclic structure. And don't confuse with the aromatic one, they are different. And the proline and the histidine, these are the heterocyclic amino acids. Another classification is hydrophobic amino acids, and the subclasses are aliphatic side chain, aromatic side chain carriers, and branch chain amino acids, and these are the hydrophobic ones. Hydrophilic amino acids, which there are two classes here, uncharged side chain carrying and charged side chains, which are basic and acidic amino acids, and neither hydrophilic nor hydrophobic amino acid. This is singular because there is only one, which is lysine. So here, my dear friends, so we will do the classification of amino acids, or I will follow this classification for the rest of the presentation. And regardless of the type of classification, what actually this is here is what the R groups in the amino acid are. Let's start with the hydrophobic, which means they don't like water. Aliphatic side chain amino acids. So they are hydrophobics, and they carry a aliphatic side chain as an R group. And the side chains of these amino acids are hydrophobic, which means they do not interact well with water and are therefore usually found towards the interior of protein. And the first two one are methionine and the cysteine, which they carry sulfur, and methionine carry a phenol ether functional group, and cysteine the phenol group. And methionine is an amino acid involved in protein synthesis. So, actually, this is compatible with all proteinogenic amino acids. All are used in protein synthesis. Methionine is also important for the transfer of methyl groups and the synthesis of sulfur-containing compounds. Furthermore, methionine is a precursor in the formation of important metabolites in bacteria. Cysteine can form weak hydrogen bonds with oxygen or nitrogen, and which makes cysteine unique. So even it is accepted as a hydrophobic, actually it carries a weak polar group, because SH, the sulfitryl or the phenol group, can form weak hydrogen bonds with oxygen and nitrogen. This group also, the phenol group of cysteine, also allows it to dimerize with a disulfide bond. This means two cysteine amino acids can form a dimer of cysteine, and it is often found in its oxidized form in protein. This oxidized form is cysteine, and cysteine covalently binds different regions of polypeptide chains, and this stabilizes proteins and makes them more resistant to denaturation. And here is the alanine, one of the simplest one, which only carries a methyl group as an R group, and it plays an important role in many metabolic events, and as well as in protein synthesis. And it is same for all proteinogenic amino acids, the 21. Alanine also contributes to energy production in the body. It is also one of the many amino acids in the structural protein. And alanine also is a gluconeogenic substrate, which means alanine is a precursor of glucose synthesis. And the proline also classified as an heterocyclic amino acid. Actually, it's an amino acid because there's an amino group. and generally considered to be hydrophobic, and is frequently found in the folding regions of polypeptide chain, and is particularly seen in collagen, as proline, also as its modified forms. The methionine, alanine, and proline, they are little nonpolar with the R groups. But what I said for the cysteine, there is a little exception, because the SH group, the sulfatoryl or the phenol group, contributes a weak polarity for the cysteine. And here you see the cysteine pattern or cysteine configuration, which 2 moles of cysteine amino acid forms a disulfide. And it's a covalent type of bonding. And disulfide bonded residues in a polypeptide chain also contributes the hydrophobicity or the nonpolarity of the molecule. And such disulfide bonds play a special role in the structure of many proteins, especially the extracellular matrix proteins, by forming covalent bonds between parts of the polypeptide molecule or between different polypeptide chains. And these are the cysteine residues forming disulfide cysteine. Hydrophobic amino acid carrying aromatic side chain. And here you see the famous benzene ring, which makes the compound aromatic. Benzene, and the benzene ring here is the benzene ring, which is the phenylalanine, and the benzene ring also carrying an hydroxide group, which makes it a phenyl group, tyrosine, and the tryptophan carrying an indole group. A benzene ring is with another ring structure carrying nitrogenous ring structure, which is called indole group. And the ring structures attached to the carbon-in-carbon, attached to the alpha carbon with a methylene bridge. So these are the 3 aromatic amino acids. And these aromatic amino acids are important hepatic metabolites. And for tyrosine, due to the tyrosine hydroxyl group, it can interact with water and form hydrogen bonds with water. Tryptophan and tyrosine, and to a much lesser extent phenylalanine, absorb ultraviolet light. And this explains the characteristically strong light absorption of most proteins at a wavelength of 280 nanometers, a property used by researchers in protein characterization. And you can look for Lambert-Beer law, which describes the absorption of light by molecules. And these 3 aromatic amino acids are also critical or essential precursor of some molecules in body, especially their hormones or neurotransmitters, such as the tyrosine is a precursor for thyroid hormones T3 and T4, and also the adrenal medulla hormones, adrenaline and noradrenaline. And tryptophan is a precursor of serotonin and melatonin. And almost 80% of phenylalanine is used to synthesize tyrosine, and the rest is used for the protein synthesis, as well as the other ones also used for protein synthesis. And phenylalanine is an essential amino acid. This is an essential amino acid to synthesize tyrosine. And what I said, almost 80% is used for tyrosine synthesis. Hydrophobic branch side chain amino acid, the famous BCAA. And here there are 3 BCAAs: leucine, isoleucine, and the valine. And these are nonpolar aliphatic R groups. They carry nonpolar aliphatic R groups and the branch chain aliphatic R groups. And so each carries an aliphatic side chain containing a methyl group. And here is the aliphatic side chain. Also indicate the methyl substituent here, and also another methyl substituent for the valine here. And they are all essential. They should be dietary in highly organized animals. or higher animals. In other words, they are essential, and these animals lack the enzymes necessary for their synthesis. Although present in many tissues, BCAAs are catabolized intensively by muscle tissue for energy. Hydrophilic. Now we are going to pass to hydrophilic ones. We finished hydrophilic amino acid. Let's look at the hydrophilic ones, which the ones like water. Hydrophilic amino acids with uncharged side chains. This means the R groups do not carry charge, either positive or negative charge. In other words, they are neutral. So these are polar. This means they have ability to interact with water. And look at here, these ones carry hydroxide group. And here there is an amide group for these ones. So what are they? This is asparagine and the glutamine, and they have a common feature. They both carry amide group. The difference in the carbon number in the R chain, there are 2 carbon here, 3 carbon in the R group. And asparagine and glutamine are amino acids that play important roles in the functioning of proteins in the body. Research has shown that these amino acids can interact with each other in various ways, potentially affecting protein folding, stability, and activity. More specifically, asparagine has been shown to contribute to the hydrogen bond networks in proteins, while glutamine can act as an acid or base depending on the pH of the environment. These interactions may have implications on various biological processes, including muscle growth, immune function, and the nutrient metabolism. Glutamine is also particularly notable for playing an important role in intracellular nitrogen transfer and energy production. Let's continue the other ones. The ones carrying hydroxyl group or alcoholic group. They are the amino alcohols or the amino acid carrying alcoholic groups, is serine and threonine. Serine and threonine, a hydroxylated version of alanine, have an uncharged side chain and contain an hydroxide group. It therefore interacts strongly with water as a result of the formation of hydrogen bonds. So they are much more hydrophilic than other closely related amino acids. Let's also remember the serine amino acid from the topic phospholipid. The phosphatidyl serine lipid. So this means serine is also important in the phospholipid synthesis. They play an important role in various biological processes, including metabolism, cell membrane structure, cell signaling, and protein folding. And the polar group can affect the solubility and stability of the proteins and influence its bioactivity. Their threonine-specific protein kinases are particularly involved in metabolism, and these enzymes are involved in the regulation of cell proliferation, programmed cell death, which means apoptosis, cell differentiation, and embryonic development. Hydrophilic charged side chain amino acids, the basic amino acids. So these are the amino acids containing positively charged R groups. And look at here the positively charged, and also this is an heterocyclic amino acid, which is histidine. And here is the lysine and the arginine. And amino acids, the positively charged side chains include basic amino acids, histidine, lysine, arginine. Histidine plays important roles in many cellular functions. It plays both a structural and catalytic role in enzymes. In particular, histidine residues can play a critical role at the ATP binding site, actin ATP binding site, and undergoes enzyme catalyzed methylation. And histidine is also important in the protection of the methionine sheath because they participate in the hydroxylation of galactosyl-ceramide. And histidine is also important allergic reactions. The biogen histamine released by mast cells can bind to histamine receptors and trigger allergy symptoms such as itching, prostaglandin production, smooth muscle contraction, increased vascular permeability, and tachycardia. Arginine is classified as an essential amino acid for birds, carnivores, and young mammals, and a conditional essential amino acid for adults. And arginine is a precursor of polyamines and urea, which means it is important in the urea cycle. Arginine acts as a precursor for creatine, which plays an important role in the energy metabolism of muscle, nerve, and testes. It can also be involved in many biological processes such as protein synthesis, cell division, immune functions, and hormone release. Lysine plays an important role in many biological processes such as cell growth, collagen synthesis, calcium absorption, and protein balance. It may also play a role in inhibiting virus replication, and support the immune system. Lysine is a base like arginine and histidine. There are also modified forms of lysine, which are the components of connective tissue, which I will talk them in the following slides. And here are these are the hydrophilic side chain amino acids, but they're acidic because they contain negatively charged atoms. They are the decarboxylic, monoamino decarboxylic amino. Previous ones, histidine, lysine, and arginine, are the amino monocarboxylic. In contrast, here the amino acids with negatively charged side chains are acidic and include glutamate and aspartate. Glutamate and aspartate are classified as acidic amino acids because they have a carboxyl acid group. In addition, glutamate is found in large amounts in the brain and is important for learning and memory, while aspartate is found in both the brain and the muscles and is involved in energy production. And both amino acids are important for maintaining the proper muscle function and can be found in foods such as meat, fish, dairy products, and some vegetables. Until now, we discussed the 9 of the amino acids, which are the hydrophilic, asparagine, glutamine, serine, threonine, histidine, lysine, arginine, glutamate, and aspartate. And these are all hydrophilic, which means they can interact favorably with water. And they are often found on the protein surface, where the side chains are exposed to an aqueous environment. And here is the glycine, an amino acid neither hydrophilic nor hydrophobic. And according to the R group, which there is only one hydrogen, it has a nonpolar. And also it's a unique amino acid. It's a kind of joker amino acid. Because the glycine, which is the simplest of an amino acid, it can fit into many cavities and polypeptide chains and is therefore found both on the surface and inside of a protein molecule. And glycine also has a distinct feature from the other ones. It is the only amino acid that does not carry an asymmetric carbon atom. Look at here. Because there are 2 hydrogen, but the rest carry asymmetric carbon atom. For the rest, the alpha carbon is asymmetric. So they have isomers. They have stereoisomers. They have for them this carbon is the chiral carbon, which means the stereoisomers are the enantiomers. In addition to the 20 proteinogenic amino acids, proteins may contain residues formed by the modification of common or modification of proteinogenic amino acid residues already incorporated into a polypeptide chain. Briefly, there are some modified amino acids in the protein structure. And here you see the 3 of them, the methylhistidine, methyllysine, and hydroxyproline. And the methylhistidine and lysine are the methyl derivative of histidine and lysine. And these can be a component of some protein or a building block of some proteins. Hydroxyproline here is a citrus structure. which carries an alcoholic group. This is why we call it hydroxyproline. It is especially found in the collagen, the connective tissue, fibrous connective tissue protein. In the form of, there is a tripeptide sequence of this protein, which includes proline, hydroxyproline, and the glycine as a tripeptide moiety. And collagen is a fibrous connective tissue protein. It is found in blood vessels, tendons, cartilage, and bone. It is the most abundant protein in mammals. About 30% of the total body protein and approximately 60% of body weight is collagen. There are some other modified ones, the hydroxylysine, desmosine, and isodesmosine. The lysine derivatives, or the modified forms of lysine, there are 3 different. This means lysine can give different modifications: methylysine, hydroxylysine, desmosine, and isodesmosine. And hydroxylysine is the 5-hydroxy derivative of lysine, and it is found in the collagen structure. Desmosine, or the isodesmosine, are formed by oxidation and cross-linking of 4 lysine molecules. So they carry 4 lysine residues. And these are found in the structure of elastin, another connective tissue protein, especially the elastin due to its elasticity. Arteries contain more elastin than any other connective tissue type. An illustration showing the differences of blood vessels in terms of the elastin content, smooth muscle content, and the collagen content in terms of elastic tissue and fibrous tissue. And look at the arteries here, from aorta to capillary and to venule to vena cava. These are the veins and the arteries. So the arteries has more elastin content than the veins. But the other proteins, or the muscle and the collagen, there is no major difference. But compare the aorta and the vena cava, a huge difference in the content of elastin. And these are another modified amino acids: phosphorthreonine, phosphotyrosine, phosphoserine, and gamma-carboxyl. These are especially the first, these 3 ones. These are the phosphoderivatives of threonine, tyrosine, and serine. And phosphate group is attached to the alcoholic groups of these amino acids because they have a common feature: they carry hydroxyl group. The gamma-carboxyl is synthesized by the carboxylation, look at here, the carboxylation of the glutamic acid or glutamate in the liver. The phosphoderivatives of serine, tyrosine, and threonine are especially involved in the construction of regulatory proteins. Gamma-carboxylate is involved in the structure of coagulation factors, many of them, such as factor 2, which is prothrombin, factor 7, 9, 10, and protein Z. And this carbohydrate, the modified glutamate, which is gamma-carboxyglutamate, provide a chelating feature to these proteins, which is so important in the coagulation. And so the carboxylation of these coagulation factors, or to obtain the gamma-carboxyglutamate, vitamin K is required. And especially the gamma-carboxyglutamate residues, which shortly GLA residues, are needed in calcium binding and in exposing hydrophobic membrane binding regions of the cell layer. And the last modified amino acid, selenocysteine. And look at here, my friend. There was a 3-letter symbol and a single-letter symbol for selenocysteine. Selenocysteine also referred to as Se, the symbol of selenium and the cysteine residue, is the 21st, some say it is the 21st proteinogenic amino acid. And why it is called selenocysteine? Because the pattern, the building diagram is same with the cysteine, or also we can say it's a derivative of cysteine, because here instead of sulfur they carry selenium. And the proteins carrying selenium as a selenocysteine residue, we call them selenoprotein. And selenocysteine is an amino acid found in humans, plants, protozoa, archaea, and bacteria. And the seg is called the 21st amino acid because there is a special codon synthesized or special codon trigger the adding of selenocysteine residue to the protein. And some examples of selenoproteins are that we did in the bioelement topic, sulfate peroxidase, periodic synthetic. And it occurs in every domain of life, including important enzymes found in humans. And here there are non-protein amino acids. About 300 additional amino acids have been found in cells. And these have various functions, but are not all components of protein. Apart from the 20 amino acids that participate in protein structure, we have non-protein amino acids, which they do not participate in protein structure. However, they are important intermediate metabolites or precursor molecules during metabolism, such as beta-alanine. It is a constituent of pantothenic acid, which is vitamin B5. Thus, it is a component of the coenzyme A, which is the coenzyme of vitamin B5. And homocysteine and the homoserine are important intermediates in methionine metabolism. Citrulline and ornithine are involved in the hepatic urea cycle and are important intermediates in the synthesis of arginine. Remember the taurine from the bile acid. Taurine is an amino acid which is used to conjugate the bile acid and especially essential amino acids for gets. And the gamma aminobutyric acid, which is a neurotransmitter inhibitor in the central nervous system. It is mainly found in the substantia nigra, globus pallidus, and hypothalamus. Alcohols, barbiturates, and benzodiazepines increase the GABA effect, especially in postsynaptic regions. Proteins taken into body must be broken down into amino acids in order to be absorbed from the intestine. Amino acids absorbed from the intestine go to the tissues via the blood and are used in protein synthesis. And we know plants are autotrophs. They do not require amino acids for their nutrition or to get from the outside. They can synthesize. But for the animals, require many of the amino acids from out. Essential amino acids or exogenous amino acids are amino acids that are not synthesized by the organism and are required to be taken externally with food. On the contrary, there are non-essential amino acids that do not need to be taken from outside and are synthesized by the body. This synthesis is not same with the synthesis of amino acids in the plant. Actually, they are derived from the other group of molecules, especially the alpha-keto acid. An amino acid which is capable to produce is alpha-keto acid and not essential, and they can be synthesized during metabolism. So the beans contain more protein than meat, but not as valuable as meat, because beans do not contain as many essential amino acids as necessary, especially for the carnivores and for the humans, such as branched-chain amino acids. They are rich in animal, especially in the meat. So this doesn't mean the plants don't have these amino acids. Yes, they have, but the fruits of animal origins carry or contain more these amino acids than the plant fruits. And here is a table showing the essential amino acids for human, dog, rat, swine, and the chicken. And also some are especially essential for the young ones, not the other ones. And look at here, branch-chain ones, the lysine, isoleucine, and the valine. These are essential for all. And look at the phenylalanine, essential, and which is required for the tyrosine. And tryptophan, which is also required for the melatonin and the serotonin synthesis, then essential. And the lysine, the connective tissue amino acid, or the amino acid of the connective tissue proteins, is essential amino acids. The methionine, which we know every protein synthesis starts with the methionine, is an essential amino acid. And threonine here, an alcoholic amino acid, is also essential for all living organisms here. And don't forget, and look at here, almost half of the amino acids are essential. The ones which are not essential, they can be derived from the alpha keto acids, which they also yield these keto acids. Let's look at the physical properties of amino acids, and here is some basic features: the solution or the solubility, melting points, and also their flavors. Almost all are sweet, but also there are some bitter and taste, such as lysine and isoleucine and arginine are bitter. Amino acids are ampholytes or amphoteric electrolytes or amphoteric structures. Since amino acids carry a carboxyl group along with an amine group, they are both acids and bases at the same time. The alpha carbon of amino acid is at physiological pH. The carboxyl group is unprotonated and the amino group is protonated. So thus here yielding carboxylate anion or the ammonium cation. At the isoelectric point, which corresponds to the physiological PA and is abbreviated as the PI, isoelectric point, hydrogen ions transition from the carboxyl group to the amino group, which means the carboxylic acid group coexists with a negatively charged carboxylate anion, resulting from the release of hydrogen ions, and the positively charged ammonium cation, which gains the hydrogen's ion. And molecules posting both positive and negative charge, as seen here in the glycine, are termed zwitterions. And zwitterion, the zwitter means, or zwitterion means in German, hybrid ion, capable of acting as an acid or base, or they have a dual structure. Compounds capable of adopting this dual structure, forming zwitterions, are amphoteric and are commonly referred to as ampholytes or amphoteric electrolytes. And here we see the zwitterion form of the glycine at isoelectric point Pi, and to form a cation in acidic solution and anion in alkaline solution. And if an electron current is continuously passed through amino acid solution, amino acids migrate to the anode or cathode, depends on the environment, whether it's alkaline or acidic. And each amino acid has a characteristic titration curve that we can use this titration to analyze them. And also due to these features of amino acids, they have a buffering capacity. This is why the proteins are also buffers of the organism. And the Zwitterian form or the ionic state of an amino acid may differ due to the factors such as temperature, ionic strength, and the presence of ionizable groups in the microenvironment. Optical activities. Amino acids, my dear friends, are optically active, but not all of them. All amino acids, except glycine, are optically active. And remember the course of organic chemistry, the tropic isomerism and stereoisomers. Yes, for all common amino acids except glycine, carbon is bound to 4 different groups, which means the alpha carbon is an asymmetric carbon atom, and the chiral carbon due to their optical activity. So it's a chiral center. But in the glycine, remember the R group is hydrogen, which makes the carbon, which makes the symmetric carbon, not the asymmetric carbon. And there are 2 mirror imaginary forms, L and D isomers or enantiomers of amino acid. An amino acid similar to D-glyceraldehyde are called D-amino acids, and amino acids similar to L-glyceraldehyde are called L-amino acids. And due to their optical activities, they rotate the polarized light into the right direction, which we call dextrorotatory, or the left direction, which is called levorotatory. While almost all carbohydrates in plants and animals belong to the D series, amino acids in proteins belong to the L series. And here is the comparison of the glyceride and the alglyceride. Remember, they are the mirror image, and we focus on the hydroxyl group here, which is the farthest hydroxyl group to the carbonyl carbon. Actually, there must be a double line, carbonyl carbon. And here the D-amino acid and L-amino acid, and compare it with the glyceraldehyde, and put the carboxylic acid group similar to the aldehyde group, and then compare the amino group. If it's the right side, or as in the hydroxyl group here, it's in D-amino acid, and it's similar with D-glyceride, it is the L-amino acid. And here is the comparison of the L-alanine and D-alanine, and also L and D. But what are the differences? So here, the upper part is the perspective formula of the L and D-alanine, and the bottom part is the projection formula of the L and D-alanine. And in the perspective formula, the straight wedge-shaped bonds, the thick ones here, go outside the paper, and the dashed ones are behind it. In the projection formula, the horizontal bonds are assumed to extend beyond the plane of the paper, the horizontal bonds, and the vertical bonds remain behind it. And however, projection formula are often used indiscriminately and are not always intended to depict a specific stereochemical form configuration. Almost all biological compounds with an chiral center usually occur only in the D or L stereoisomeric form. For example, the amino acid residues are often found in some peptides, in bacterial cell walls, and in certain peptide antibiotics, but are usually restricted to small peptides. Most amino acid residues in proteins consist of L stereoisomers. When chiral compounds are formed by ordinary chemical reactions, The resulting product is usually a racemic mixture of D and L isomers that is difficult to chemists to distinguish. However, for a living system, the difference between D and L isomers is so significant because the amino acids in proteins must be stereochemically consistent. The formation of stable, repeating structures in proteins usually requires that the amino acids that make them up come from a single stereochemical series. This enables cells to synthesize L isomers in particulars, because the active sites of enzymes are asymmetric so that the reactions that they catalyze are stereospecific. Chemical properties of amino acids. And there are three types of functional groups in the amino acid molecule we know very well: the amino groups, carboxyl groups, and the R groups. So the chemical properties of amino acids depends on these three groups. Reactions of amino groups, I would like to explain them briefly. And the first one is an activation of the amino group with alcohol in alcoholic environments. Amino groups do not change the ammonium cations. Therefore, they are not amphoteric in alcoholic environment. Solvent titration, it is the method to determine the carboxyl group in the amine acid solution. It is used to determine the amount of amino acid in a solution. The bonding of the amino group with aromatic amino acids. So amino acids combine with aromatic acids to form insoluble substances. Thus, harmful substances are removed from the organism. The beta-ene reaction. When amino acids are in this withdrawal state, beta-enes are formed when three methyl groups replace three hydrogen in the amino group. The uramine acid reaction. If a urea molecule is added to the amino groups, the uramine acids will form. It is used in the identification of amino acids. The ketopiperazine formation, the ethyl esters of amino acids are condensed to form anhydrate ring structures bearing two amino acid groups, resulting in the formation of the ketopiperazine molecule. Salt formation, amino acids can form salts with heavy metals and easily crystallize, especially the salts formed with copper are easily crystallized, and this property is used to obtain pure amino acids. Esterification, if they react with alcohol in the presence of anhydrous hydrochloric acid, they will bring their esters, and the esters of amino acids are utilized in their functional distillation. Carbamino acid reaction. Carbamino acids are formed by binding carbon dioxide to amino group. When heated, it breaks down again into carbon dioxide and amino acid. And hemoglobin forms carbamino acid, which is called the carbamino hemoglobin, with carbon dioxide, and is important in the transport of carbon dioxide from tissues to the lungs. In hydrogen reaction, all three amino groups give this reaction. This means free amino acids, peptides, proteins all give this reaction. And ninhydrin is an important qualitative determination method for amino acid. It is sensitive to less than 1 gram, of great practical importance. Nitric acid reaction. When amino acids react with nitric acid, the nitrogen of the amino group passes into the free element such as nitric acid nitrogen. On the other side, the amino group is replaced by hydroxyl group. At the reaction, a molecule corresponding to each amino group is released to nitrogen. For this reason, nitric acid reassignment can be used to determine the amount of free amino group, and this reaction also called Vansky method. Chelating amino acids with metal ions. Amino groups form complex chelates with many heavy metal ions such as copper, cobalt, manganese, and iron. And here is some of them: cysteine cobalt complex. Chelating amino acids with metal ions involves the metal ion forming a complex with amino acids by forming coordination bonds. And this process can serve a variety of biological and chemical functions by forming strong and specific bonds with amino acids in the vicinity of a metal ion. These bonds are usually based on electrostatic interactions between the positive charge of the metal ion and the negative charge of the amino acid. Chelate is a complex molecule coordinated by bonds located around the metal ion, cysteine here, cobalt and cysteine. The chelation of amino acids with metal ions is important in various biological functions such as transport, storage, and utilization of metal ions, especially in biological systems. These complexes can regulate the position of metal ions in the cell by preventing metal ions from entering into uncontrolled reactions within the cell. Reactions of carboxyl. The carboxylation of amino acids, and it is a name given to the separation of carbon dioxide from the amino acid molecule. And here the D refers to the elimination of carbon dioxide from the carboxyl. And amino acids are decarboxylated in tissues to form amide. And examples include histidine converting to histamine, lysine to cadaverine, ornithine to putrescine, tyrosine to tyrosine, and tryptophan to tryptamine. Additionally, these conversions can be facilitated by anaerobic microorganisms, particularly within the context of microbial metabolism in the gastrointestinal tract, often referred to as the spoiled gut. Amines such as cadaverine and putrescine produced through decarboxylation are prevalent in malodorous meat. Despite their pungent nature, the toxic effects associated with these amines are generally low. It is crucial to recognize that the toxins associated with these amines predominantly arise from the metabolic activities of the microorganisms responsible for their synthesis. And let's look at the pattern of the carboxylation of an amino acid. Here you see the amino acid and the amine by removing a carbon dioxide from the carboxyl. And the enzymes catalyze this kind of reaction are called the decarboxylases. And here example of histidine decarboxylase, which convert histidine to histamine. It's biologic amine. And the reaction of R groups, and they are variable or so diverse. The R groups of amino acids are composed of multiple variable groups. And due to differences in the groups, in the R groups, different reactions occur. Especially they give unique color reaction. As in the Millian reaction, for example, here the Millian reaction, a red color occurred after the reaction with the amino acids carrying phenyl groups such as tyrosine and phenylalanine. This means these color reactions are amino acids to be separated and recombined. And also there are the other ones here. Yes, my friends, so we complete the topic. You can visit the website biochemia.wet to download topic as a PDF document. And here with the reference for you for further reading and study. The next topic is amino acids, peptides, proteins-II. We study part 2. And also, you can also visit my website for more about biochemistry. You can follow my Instagram account. And my dear friends, please don't forget to subscribe to the YouTube channel, to like the videos, and turn on notifications to be informed about the new videos. See you in our next topic. Goodbye and stay safe.